All right. Good afternoon, everyone, and welcome to Lincoln. I'm a local person on the committee. So I'm a plant physiologist, and my interest is in understanding how crops, especially cereals, respond to abiotic factors. Two primary factors that I work on are while evading a fly, the heat stress as well as drought stress. So today I'm going to talk about some of the work we have done in terms of looking at the dynamics of how crops or plant responds to a particular stress. So the dynamics, I mean both the temporal as well as the spatial dynamics, with the idea being that the plant typically for most stresses would not respond uniformly and would not respond in the same way consistently. It would evolve with the stress and the time. So the work would be on rice, and the idea that I want to pursue is higher temperature. As you can see, this is the historic data for how the temperature anomalies have played out at the daytime highs as well as the nighttime highs for average. So in red is what you would call is like higher than the average or temperature, so anomalous temperature, and the areas that are blue are the ones that typically have kind of been colder than what would be an average over the almost 100 years. The two key things I want to point out here is that there's a lot more nighttime increase in temperature as opposed to daytime. So because there's a lot more red that you see on your right as opposed to daytime. The second thing that I want to point out is the daytime highs seem to be very speckled or sporadic in terms of spatially, whereas the nighttime increases seem to be more widespread and more continuous. In the U.S. itself, in the last 100 years or so, the nighttime temperatures have increased at twice the rate of daytime temperature. So understanding, how plants are specifically responding to nighttime temperature is quite important. The crop that I'm going to talk about is rice. These are a little bit dated, but they show what in terms of kilocalories per capita is consumed globally. So if you combine rice and wheat, it collectively feeds more, provides more than 50% of the total calories that we consume as a species. All right, that didn't go well. So this was data from 2004 from field experiment led by the International Rice Research Institute, which showed that for about 1 degree increase in the nighttime temperature or the average low temperature during the growing season, you could get up to about 10% decline in the yield. So the matter is not only about the yield, which certainly is very, very important, but it's also about the milling quality. So rice needs to be polished, and if you have milling issues or quality issues, then the quality, in terms of breakage and usage, you have poor rice, and a lot of it can get wasted and not consumed by humans. Similarly, hybrid rice, there's some evidence that suggests that hybrid rice, which is the type of rice that grows a lot in the U.S. And in many other parts of the world, is also more susceptible in terms of quality and yield to higher nighttime temperatures. For my study, I'm going to call high night temperature, since it's a long word, HNT. So from a physiological perspective, we decided to focus on the reproductive stage. As the temperature increases at nighttime, the rate of respiration increases. So plants start to lose more of the carbon. During vegetative stage, there's ideas and some evidence that plants are able to compensate because they're actively photosynthesizing, and they've got more carbon than normally they would have during reproductive stage, where they're trying to actively store. So it also not only you lose carbon, but the carbon that is packed, which is starch, in primary form of starch in the rice endosperm, its rate of accumulation and how it is packaged is disturbed, which means that the seed actually becomes a little bit weak. So during milling, it could break more easily. So when we study rice grain quality or chalkiness, we talk in terms of the rice panicle architecture. So there are grains that are pretty nice and plump, and that are called superior spikelets. So these are all branches, and these yellow blobs are grains or spikelets. And then there are grains that look a little bit half-filled, not so small and like a little bit off types. So these are called inferior spikelets. So that creates a pretty interesting challenge. You want a lot more superior spikelets, but higher nighttime actually gives you a fairly decent, significantly more inferior spikelets. Given that, what we wanted to explore was to characterize this response, but then do it for a large number of rice varieties from a diverse germplasm that represents a diverse germplasm. And the reason for that was that we wanted to understand if there is, what's the genetic basis of this response under high night temperature when the grain is developing in those conditions, so that we can come up with genes and markers that could help improve that trait. So to do this, we set up an experiment, to do this, we set up an experiment where we took about 300 rice accessions from one of the several diversity panels that people use in our community called the Rice Diversity Panel 1. It represents about rice from all major subpopulations as well as from 80 countries. So we used this and we grew the plants under controlled condition in a greenhouse, and then depending on when half of the primary panicle, which is the first panicle to emerge, is mostly flowering, we mark those panicles. We wait for a day because that's the day when we mark it. When it looks like this is the day when it's fertilized, we let it go for one day, and then we take a subset of our plants from that particular accession, and we continue to grow them in controlled conditions, and we also take the other part into a high night temperature greenhouse where they mature under that condition. So essentially, it's 30-degree daytime, 23 for night, and it's 30-degree daytime, but the night temperatures are increased by 5 degrees, so between 4 and 5 degrees given the greenhouse's control. And then what we do is we wanted to capture the dynamics of the panicle development. So we imaged the panicles 3 times, so week 1, week 2, week 3, depending on when they fertilize. And it's a diversity panel, so it goes anywhere from 2 months to 4.5, 6 months, but we capture the reproductive stage. This is what the animation looks like. Essentially, we built a box from the local hardware store, have cameras, and a desk height adjusting mechanism from Amazon. So you could take the plant depending on its height and image this panicle. And the idea is that we should be able to capture the phenotypic variation to identify eventually superior spikelets and inferior spikelets, which obviously are not distributed. So there's a lot of heterogeneity from that perspective. So this is what the real thing looks like. The rest of the plant is underneath. So we just have the panicle, and there's cameras that go around at different angles. And we use these images then to make a 3D reconstruction. So essentially, if you want to look at this, we have week 1, and we have week 2 and week 3. So as it progresses, the volume changes, the surface area, and the color, all of those things change. So we used, let me see if the animation would work. So we used the NERF software to make 3D reconstruction. So this is not an image, it's a 3D reconstruction, and it comes out very clean. So we can use this to capture a lot of phenotypes, like related to color and volume and morphology. One instance to show for a handful of genotypes, this is just the ratio of green pixel to red pixel, which would kind of be indicative of maturation. you could see that they're week 1, they're kind of similar. Maybe the HNT, which is the light gray box, is kind of exceeding, but not significantly. But then you see that progression, so it advances much faster and then looks different at the end of week 3 at the whole panicle level. So we next asked if, since we have this panel and it's heavily genotyped, so we have about 1.3 million SNPs for this, could we then do GWAS and see what do we find? So we used, we used the week 1 data for just the green pixels. And one of the major peaks, these are just 12 chromosome of rice, and that kind of caught our attention was a SNP that lies in the in a gene called SPED1. Several years ago, a group in Jiang et al. Had shown that if you mutate SPED1, you get these very small branchings or rachis, so that creates a very compact panicle as opposed to a wild-type panicle like this. So this kind of showed us that what we were observing is actually, capturing the genetic variation, and there's like a, validation for what we are doing. So the next thing we wanted to ask was, we were interested in this superior and inferior, and we know that they're not uniformly distributed. So could we actually segment? So if we have a 3D volume, like a 3D point cloud structure, could we actually segment or slice it into 10 geometric segments and then treat each of those segments as a independent or as a trait? And could we then analyze that to see if we could find the basis for this, differences that may be spatially, correlating? So to do that, so just to give you an example, this is what it looks like in week 1. So it's very compact, not filled. As the grains get heavier, they get more plump. They start to spread out a little bit more, and then eventually, as they mature by week 3, they are like changing color. So this would represent the red to green ratio that I was talking about. So we did that for the phenotypic data, and we also ran a genetic correlation analysis for these using the red pixel count for control and HNT stress. So this is a heat map which shows that what's the correlation between each of those traits. So if each of those slices next to its next slice and every other slice of that panicle. So what we find is that there's definitely some strong correlations. If you have a positive correlation spatially, you would see this in red, and if it's a negative strong correlation, you would see this in blue. And what we find is that the traits or the segments that are next to each other are genetically correlated. What that means is that there are genes that are commonly shared in regulating that particular trait. In this case, that trait being a digital trait, basically a red pixel count for that segment and the next one. We also found that the controls have a lot more well-partitioned. So there's a clear middle and there's a clear bottom and a top part, because these are kind of correlated, these are negatively, and then these are again positively correlated. So that seems to erode under HNT stress, which is to be expected, because I think as the stress continues, the HNT accelerates the maturity of the panicle, so you would expect something like this. So this analysis was done by my collaborator at Virginia Tech, Gota Marota, and his lab group. So with this idea, we actually did GWAS for all the normally distributed characteristic that we could find for different pixels and ratios and volumes. And so there's an instance of what one of the results looks like. So you could have, in this case, you could have a strong peak at in segment 4 and segment 5 and 6, but not in 7. So that shows that there's a genetic basis for how the panicle is developing in the different temperatures that has a spatial basis to it. So you may not see it if you were to just image the top of the panicle or the bottom. We ran this analysis, so I want to give you and we found several very significant SNPs. So I'm going to give you a snapshot of what we did with one of the SNPs. So this is what we call HNT 5.1. So this is the hist, the third segment, and our red pixel under control. And this is the third segment from the bottom, and the red pixel under HNT. As you can see, one of the major peaks here, which we call HNT 5.1 on chromosome 5, has a strong signal here, but it's missing completely from control. So we investigated the gene, which is in proximity of this particular SNP, and we find that this gene is not characterized in rice, but it has a very strong diurnal regulation. So I want to pause and kind of mention that plants perceive day and night through light and temperature. So if you make the nights warmer, you would think that it would perturb some of how they perceive the beginning of the day and night. So so this gene actually peaks quite strongly around 4 a.m. and 6 a.m. and then it kind of goes screech down, and then it starts to go back up and peaks again. So this is from a public data set collected in the field over a development window. So we selected one of this gene and we made gene edits. It's a FBOX gene, which is like there's several hundred members of this gene family in rice and in other plants. And we called it FBOX 5, lack of any creativity there. So what we did was we made several edits, and I'm showing you data for 3 of them. When we grew them, after a couple of generations, we find that the panicles were shorter compared to the wild type, which is shown here as KIT, and the mutants, the edits are shown here as PY, and different. So they have different types of mutations to make these 3 mutants. We then imaged them, these panicles, under control condition and under HNT condition in week 1, week 2, and week 3. We detected this SNP on week 3 panicles, and what we find is that the only place that we see a difference in the R to G ratio is in week 3 and under HNT. So if you look at just the controls that we see, they're not at all different, at least not very significantly. But if you look at HNT, you have a wild type, and then you have these mutants. So they're kind of diverging, and the divergence is kind of starting around that segment 3-4 area. So that kind of is one example, and we have one more of that. But we wanted to explore this a little bit further in terms of we don't know anything about this gene, and we think it's kind of important. So we looked at the expression of the mutants, 2 of the mutants, and the wild type at 4 a.m. and 6 a.m. Red being the HNT and blue the control. We see that this gene is suppressed in the mutants as well as the wild type by HNT conditions. When it's supposed to peak, it doesn't peak as much under high night temperature conditions. So when we looked at the other genes in the genome through RNA sequencing, we found that some of the major clock regulating genes like TOC1 and PRR37 at Gigantia and another clock gene CCA1, they are strongly disturbed by this mutation. So the first half is basically the wild type, and then you have the mutants at 4 a.m., and then you have the wild type again at 6 a.m. and the mutants. But at 4 a.m. especially, these 2 genes that are sort of central for how the plants know that it's daytime versus nighttime. So this gene seems to be upstream of that clock, of these clock genes, and obviously their regulation is disturbed when you look at the second half of this 4 a.m., 6 a.m. under HNT conditions. So that kind of gave us some new clues to how the warmer nights may be regulating the high night temperature response. Just kind of before I conclude, I know it's a gene bank talk. So the different allelic forms, which is like the major and the minor. If you look at how the the 300 lines are distributed, or the ones that have one form of the allele or the gene globally. This is what it looks like. It goes all over pretty much. But if you look at the minor alleles, their distribution is mostly at high latitude. There's hardly anything in the major, some of the major rice growing regions. So that sort of suggests to us that this gene is actually important for potentially like latitudinal regulation, and that higher nighttime temperatures. When we think of higher latitudes and lower latitudes, we think mostly in terms of light and photo period, but perhaps the nighttime temperature is also a factor in there. I know my time's kind of running, so I just want to summarize a few things. One, that you have conditions we can screen for stress tolerance, but there are loci that are very unique, as I've shown, to the stress conditions. And if you're screening germplasm and material under optimal conditions, you may not detect them. So this would be like one or at least a third instance that my lab's kind of shown that in case of temperature. I also want to point out this idea of 3D reconstruction. I think not that I'm pointing out, I'm reinforcing the idea from the talk, the talk before mine, and several other talks and posters, that, this 3D is not just like a fancy, oh wow, look how nice the pictures look. There's actually real potential to understand the dynamics and variation. And so, there's a lot of promise here. And finally, I think we've looked at this gene in corn and sorghum as well. It's not characterized there. It also has a very strong circadian regulation. So we're starting to study that to see, if we could find more evidence for its adaptation in terms of wider germplasm. And with that, the most important slide is, if it would advance, I want to thank my current and past lab members, especially Anil Chandran. This won't be possible without my computer science collaborator at UNL, Hong Feng. And a lot of work was done on the genetics side by Gota Marota at Virginia Tech and his lab. And with that, thank you. And if you're interested, we're looking for a postdoc who could work in this area. So please feel free to reach me. Thanks.
Thank you very much, Harkamal. I want to open the discussion now. Any questions? Everybody is looking forward to the coffee break. No, we have a question here.
Great. Thank you for your great talk. I didn't know even that such small variations in night temperature could have such an impact on the physiology. So I was thinking if did you do any typical physiological measurements at the beginning of the day, like stomatal conductance, photosynthetic efficiency, to see how those plants get up?
Yes. So so in terms of the conventional phenotyping, we did measure the grain parameters, so grain length, grain width, grain weight, and we found and validated the genes for those. None of those hits match with what we are seeing with this dynamics. In terms of the physiological measurements, it would be very difficult to do this for several. I mean, we're talking 1,800 plants, so it would be very difficult to kind of have a physiological traction for doing this on a dynamic basis. But what we did do is we took some of the mutants and wild type and some of the diverse lines, and we are looking at or we're doing experiments where we can look at the panicle level gas exchange under different temperatures. So we have a customized cylinder that we built with Leica, and we combine that with the imaging data so we could get the surface area to normalize, because every panicle is going to be different. So we can attach a 6800 to that cylinder of living plants and have different temperatures. So we are doing some of it, but that's just completely intractable as far as I can envision with a genetic diversity site. But I'd be happy to show you what we have so far. That's a nice question.
Thank you.
Thank you so much for the talk. It was quite interesting.
Thanks.
I was curious about the 3D reconstruction. You said that's a very important new tool. And what I wanted to ask was, and you used it in a way I hadn't seen done before. I was curious what you saw as being like the future of 3D modeling and what are some things that you would like to do in the future with it?
Okay. I was wanting to show that in this presentation, but somehow it did not update on my, so we built this system. Essentially, the whole system with cameras is probably going to be cheaper than the 4-night stay in this hotel. The new NVIDIA software called NERF, if you look it up, would not require the 120 images we use to analyze this. So that software can do a much faster job on a less computing heavy setup with a simpler camera and maybe 3 images rather than this whole 1 a second from 2 cameras. The second thing I wanted to highlight, and I could show that, sorry about this update, but my colleague Hongfeng Yu developed a graphical interface for this. So that interface, he's going to basically he's fine-tuning it, but essentially you can upload these images and it can analyze, and then you can look at different segmentations. You can track different branches over time if you have the images, and branch length and seed number, and individual seeds can be tracked for basically a month. And in terms of the gene bank enhancement, that's like 500-600, and an open publicly open source software that we are developing and want to upload fairly soon once they publish it in a conference. Computer science people do conference publications. So once that's done, it's a powerful tool and very cheap, as long as you have hands to work through to kind of be gentle with those plants as you grow them, whether it's under control conditions or not. So yeah, so that's definitely maybe I rambled off your question a little bit, but I wanted to say that because I had it, but it didn't update. But yes, I can show you if anybody else is interested. I have an updated copy of my presentation on my computer. So
I want to close discussion here now because we are running a little bit behind schedule. So in order to allow a little bit of a coffee break, I see you back here in about half an hour. One more applause for Harkamal.